Okay, so these are the tools. So there are 4 altogether. RE, or restriction enzymes, so restriction endonuclease. And then we have the cloning vectors, another one host, and ligase. So we're going to take a look at each one for the tools in recombinant DNA technology or genetic engineering technique. Okay, first tool in genetic recombinant technique or genetic engineering is restriction enzyme or RE. So this RE is actually the function, if you look at the scissor here, is to cut. Other word for cutting is digesting, enzyme to digest, or enzyme to degrade, degradative enzymes. You can use this word as well. So cutting, digest, degrade. Another one is cleave. You can use the word cleave as well. Or the first stage of gene cloning, which is the isolation of vector and gene source DNA. So first, you need to isolate the source of DNA, example like human tissue cells grown in lab culture. Another one that you need to isolate is a factor. In this case, you're going to isolate plasmid, which is extracted from a bacteria known as E. coli. So there are 2 sources here to isolate, the source DNA and the vector, which is the plasmid. So that's the first stage of gene cloning. But before that, we look at the structure of the plasmid. So the plasmids are engineered with 2 useful genes. That's important genes that you have to take note. First is the ampicillin resistance, which is the antibiotic resistance over here. Antibiotic can have more than 1 antibiotic resistance for the plasmid. So this ampicillin, this one how you put it is different now. AMPR. This confer resistance to antibiotic ampicillin. Okay. Another one is the LAC-ZAC area here, the red one here. This is the LAC-ZAC genes, the LAC-ZAC genes that encode the enzymes beta-galactosidase that catalyzes the hydrolysis of sugar. Yeah, the big LAC-ZAC. And here as well, there's a restriction site. Restriction site is only located at LAC-ZAC area, which is the red one here. So you can see that there's different types of restriction enzymes. You have EcoR1, SMAL1, BAMH1, and so forth. So there's a specific site of cutting at the LAC-ZAC area. So restriction site is just one, okay, for each of the specific RE. So plasmid has single recognition sequence. So take note, single. Satu je. But for human DNA source, it has sequence, banyak. That's why for human, it will produce many fragments. But for plasmid, just one site, one restriction site. So plasmid, single recognition sequence within the LAC-ZAC gene, within here, for restriction enzyme used. Okay, next stage of the gene cloning is DNA cleavage, or cutting of DNA. So both the plasmid as well as the DNA source need to be digested or cleaved with the same RE restriction enzymes. So for the plasmid, it needs to cut here. For the plasmid, it needs to cut at single restriction site, which is located at LAC-ZAC area. Remember that LAC-ZAC codes for enzyme beta-galactosidase that breaks down lactose into glucose and cuts the human DNA at many sites. This is what I've mentioned before, and you'll generate thousands of fragments. This is example of foreign DNA, so it has multiple sites, okay, or many sites of enzyme, restriction enzyme. So you're going to produce a lot of fragments here. So some of the fragments that carry the human gene of interest, not all fragments that carry gene of interest, because you have to take note. That's why afterwards, going to have screening methods, and then after that, we need to have a method which to identify the gene of interest. It's going to be another technique, which is known as nucleic acid hybridization technique. Nucleic acid hybridization technique is used to identify the gene of interest. So take note on that. So all the fragments have complementary sticky ends. So this is the steps. The restriction enzyme is used to cut a specific DNA strand from the DNA of a cell. Okay, so this is the cell, and this is the DNA from the nucleus. So extracted the DNA from the nucleus, and we have this. They're going to have restriction enzymes that cut the strands. And here you can see that the few sites of RE, not just one site of RE. So it will produce multiple fragments of DNA. So this is the cutting of a plasmid at specific restriction site or single recognition site, which is located at the leg sac area. So this cutting of the eco R1. So restriction site is between G and A. So you need to cut at both strands of DNA. One strand, so cut the GA, another strand, you have to cut the GA, and it will produce the sticky ends like this, which is staggered and incomplete. This sticky end is single-stranded DNA, which is then can pair or complement with another DNA from another DNA source, okay, and produce the recombinant DNA. So this is a DNA source, you can see here, and cut with the same EcoR1 or same restriction enzyme, same as with the plasmid. And here, we're going to produce the sticky ends like this. So this will be attached to the plasmid and produce recombinant DNA.